Welcome back to yet another journal club brought to you by Lifespan.io. I'm Dr. Oliver Medvedik, and happy belated New Year to everybody. I think this is the first show we have of the new year, right, Steve? October. What month am I in? January. Think about Halloween, January 31st. We have a short paper that we're actually covering today. I always say that this is going to be a short paper, and then we end up rambling for 2 hours. But this is a short paper that came out very recently, and there was like a paper that came out right on the heels of this, or I don't know exactly if they came out at the same time or maybe a few days apart, but David Sinclair's lab, the lab that I did my PhD thesis in, came out with a very similar, much more expansive paper in Cell, which maybe we'll cover next, or maybe there's always exciting stuff. So I know Steve mentioned there's a collagen paper, and Orrin brought up a paper as well to look at. So we've got so many papers to cover, but this is a paper that's very intriguing and kind of I don't know if I could say this is what the field has been like waiting for, but it's a paper involving Yamanaka factors and using them to extend the lifespan of normally aged mice. And is DA is here with us. Great, DA, you're always asking, did they look at the lifespan extension of the organism? And in this paper, yes, they did. So finally, we can deliver on that. So usually there's a lot of stuff that kind of floats around, and they don't really get around to doing the lifespan extension stuff, but they did indeed in this paper. So that's one of their key figures. So the paper, unlike most of the other papers that we've done, which come from academic labs, this is from a biotech lab. It is Rejuvenate Bio from San Diego, Massip et al. Gene therapy mediated partial reprogramming extends lifespan and reverses age-related changes in aged mice. And this basically came out in, how do you pronounce it? BioRxiv, I guess, right? BioRxiv. BioRxiv. So this is like not gone through the official peer review process, but I think it basically came out in this format really quickly because I'm going to speculate that maybe David had a ton of stuff that he was publishing and nobody wants to get scooped, so all of this stuff came out at once. So apologize for the sirens out there. We're in New York City. City's always exploding, so I'm just gonna wait for that to pass. Yeah. So let me get back here. All right. So Rejuvenate Bio is the company, and I believe it was co-founded by George Church as well. I was kind of looking through the who's running the organization. George is on our scientific advisory board. So and we also track Rejuvenate Bio if you're interested in seeing what they're up to on our website as well. So I don't think they were necessarily focusing on this technology per se, but they're looking at other related technologies. Let's take a look here. Okay, I'm going to share my screen. So what are the reprogramming factors? Well, it's the famous Yamanaka factors, right? Or a subset of them, as they mentioned, OCT4, SOX2, KLF4, OSK. We've covered papers in the past where people have used cellular reprogramming factors to tackle certain aspects of aging. I believe we looked at a paper where it was employed in mice that were prematurely aged. I can't remember if these were the lamin A mutant mice. We also looked at cellular reprogramming factors to, get rid of senescent cells, but nobody's really looked at these factors. As I think this may be the first paper, as well as David's paper that just came out, although I haven't read that one yet. This might be the first paper where they've actually introduced these cellular reprogramming factors into normal aged mice and saw what happened, and actually got a significant increase in lifespan, as well as a decrease in frailty index. So So let's take a look at what they did. The cool thing about this paper also is that these weren't transgenic mice; these were normal mice, and they used these AAVs, these adeno-associated viruses, to deliver the system into these aged mice. So there's a couple of cool points here. Let me just scroll through here. Everything's on the. This is just background. There we go. So they use this kind of dual AAV system where transcriptional activator is turned on by adding doxycycline, so an antibiotic, to the water, and that switches on this other vector, which expresses the Yamanaka factors. And I don't know, I don't work with mice, so I don't know where's the optimal place to inject these AAV9 vectors. Maybe somebody can comment, but it's a suborbital injection, so right behind the eyeball. That's where they do it. Usually they do it, sometimes I hear them doing it intraperitoneally, but here they do it this way. So Mika, you've got a comment.
Yeah, I was curious about that as well, so I looked it up. Apparently this is just an easier way to get it into a vein in a mouse than the tail, apparently.
Got it. Yeah. So yeah, I remember other people working with mice, David's lab, and usually they do tail injections, and I guess this is easier. Anyway, behind the eyeball injection, it is. So the other cool thing about this is that these aren't transgenic mice. So this application here is already kind of like one step closer to being used as a therapy, and it's not being introduced to mice that are like really young. They're actually injecting these mice when they're like 125 weeks old. So this is all the way on figure B. So these mice are like, they don't live much longer, right? I think their maximum here is 165 weeks. So these are the equivalent of, like they mentioned in the paper, something like somebody being in their mid to late 70s getting this type of injection. And here are the lifespan curves. So they have a control mice that are just basically fed doxycycline, N equals 20. The blue line is the mice that have been injected, and they also have a historical lifespan curve that I don't know exactly where they got it from, but this is the same background of mice. This is basically just to show that the mouse strain that they were using in this study wasn't like messed up in any way, I assume, right? So it's not like you're getting an extension because the mice were prematurely kind of, aged somehow, and you just boosted it back to normal levels, right? So you're actually, so the, Mika, do you have a question, or is it just?
I was curious about that, the Jack's historical. So I'm assuming that's the Jack's laboratory historical, where all these mice come from. I'm surprised they only had 33, because Jack's lab has had thousands of these mice in studies over the last I don't know how many years. I'm guessing they just pulled that from like one study.
Yeah,
I'm surprised by that number. I would have expected Jack's historical N equals 1,000, not N equals 33.
That's true. Although this may have come from like one study, right? So I don't know. I don't know how messy it would have made their statistics if they took like 100 studies, each of them N equals 20 to 30, and then just averaged them all together, right? Versus just taking just one curve that they picked out of all those studies. But be that as it's made, it was nice for them to include that. And you see a significant, like they mentioned somewhere, using the numbers that they have, correlates like 105% increase. But you can see the curves right here. 50% survival curve is quite significant, starting at the right around the time of injection, and a bit of an extension also for maximal longevity as well. The cool thing for me here as well is that this also isn't apparently optimized either, right? So I mean, I don't know how many trials they did, or if this is the best data out of how many mice they did, right? I mean, the issue with, I'm going to stop sharing here. The issue with obviously these OSK factors, as they mentioned in the paper, is that you can get teratoma formation. You can get a lot of unintended effects, right? Because these are cellular reprogramming factors, and they're quite potent. I'm quite pleasantly surprised that they get this extension by injecting these factors using this AAV system in these mice at such a late age. That's extremely promising. So that's just, I'm just going to toss that out there. Anybody else want to comment?
So that they have another curve, I think later on, or maybe it was supplements. Apparently, they started the treatment after half the original cohort of mice died. So they started with twice as many mice, waited for half of them to die, so they knew they were hitting their median lifespan. And then they started suggesting this
right here
definitely is something that can be applied very late in life. Like these mice were on the down.
Exactly.
Cohort.
Right.
So yeah, I agree. It's very, very interesting.
Yes, and then you get this nice little bifurcation there, which is really, really cool. Right, that that it's kind of nice twofer there, right? You have a system here that's already kind of being geared up for to be utilized in a therapeutic manner, and also being delivered to really aged mice as well, right? So anyway, so histograms of remaining lifespan in weeks after injection, right? So significant increase, in kind of average weeks remaining. And they also look at a frailty index. I haven't had a chance to take a look at the frailty index for mice, but basically this is a number, a score that I think it looks at a number of different physiologic parameters, right? Like gait and other types of, attributes that will tell you whether or not somebody's frail, right? Somebody's kind of hobbling along versus somebody who's much more robust and resilient. And on kind of the scores of the frailty index, the mice had a significant improvement as well. Then they switch to, so they also look at a methylation clock. I haven't looked at the exact clock that they use, DNA methylation clock, which they looked in liver and heart tissue. This is from the mice as well. Basically illustrating that, at least through the use of this particular clock that they had, you had at least from the methylation levels, kind of a more youthful state in the liver and heart. And also, they applied this technique to human cells. These were skin cells, biopsy cells from, I think, a 65-year-old individual, keratinocytes. This was done in vitro, obviously. What they didn't inject this into this guy's scalp. They just basically took a biopsy and grew the cells. And they used I'm not sure if they used, what did they use? Not AAV, but lentiviruses. MOI is, I believe it's multiplicity of infection. So that basically, MOI of 1, I think, is you've got the same amount of viral particles as cells. MOI of 0.5 means you've got half as many. And I was just kind of curious. I looked up these numbers, and it's kind of like on the low range, but it sort of varies depending on the cell type you use. So it's sort of, people do this with trial and error to maximize expression of, to get the genes into your cells. Mika, yes.
So I think something is worth talking about on the liver and heart. So AAV is well known to target liver and heart very heavily. Like I think 70 or 80% of AAV hits liver and heart. And so they chose those for the methylation clocks, which kind of makes sense. They wanted to see what they actually hit. But I think when it comes to translational medicine for life extension, keep in mind that there's a good chance the AAV completely missed entire organs, which I kind of suspect is what potentially led this to not be even more beneficial than it could have been, because if the mice didn't die of heart disease, but they did die because their lungs failed or something. And it would have been nice if they would have done some methylation clocks on other organs that are known to not be hit by AAV to see if there was any side effects. Where the AAV probably didn't do it, but just general rejuvenation maybe did. So that was unfortunate, I thought.
Yeah, that's an excellent point. Again, I don't know. I can't speak for the researchers to, as to why they didn't do that or why they, published in this format. I'm going to speculate that it was probably because they might not have wanted to be scooped based on other work that was coming out at the same time, and obviously they were doing this,
possibly
doing this for quite a while, right? So at that point they're like, okay, we've got the data, let's publish it like ASAP. But to your point there, I might, hopefully I'm not wrong with this, misremembering this, but I remember there was a paper. I think it was the laminopathy paper with mice. I think we covered that here a while ago, and I think they also used an AAV vector system. And I think one of the major organs that was missed was the gastrointestinal system. I think the intestines were for some reason the AAV vectors didn't really infect those cells. Then, so the mice they had an extended lifespan, but they died of intestinal abnormalities, which were kind of on par for not having that fixed. So so to your point, yeah, if there are major organ systems being missed by this vector, right, then you're getting a suboptimal dosage. But despite that, you're still getting a lifespan extension, which is still impressive, right? So there's clearly a lot of room for improvement, but nonetheless, it's still pointing in the direction that this is a valid therapeutic approach. So anyway, at least looking at this predicted age skin blood clock, which is also, I think, based on a methylation clock, you get also this indication in human keratinocytes that you have that this potentially will be working in human cells, as you would expect, right? I mean, it's a mammalian system. And that's it. This is basically just supplementary data telling you that you're getting expression of these factors in the keratinocytes. So they basically do a Western blot to just validate that their lentiviruses are working. So it's a very, very short but very promising results type of paper. Unlike some papers where you have a ton of stuff in the kitchen sink and you're like, well, not really sure. But yeah, Oren.
Yeah, I just wonder, like, the thing with the keratinocytes. I mean, I guess people have done similar things before, right? And it's not very related to the mice experiment. So I'm not sure what really they're trying to say here.
With the keratinocytes, you mean?
Yeah, it's not the same vector. It's the same method for delivering the Yamanaka factor. So I don't know. It's a bit out of place. Yeah,
I'm thinking again based on they had some really extremely promising data with the mice. That's sort of like the punchline and the conclusion were right there. And there was just a lot of sort of and unlike other papers, right, where you have a ton of background stuff and then the punchline is kind of lacking, you have a very strong bit of the final data here and kind of a lot of supporting stuff that we'd all like to see kind of missing. I'm thinking that they probably, they probably were in a rush to publish, right? So to get this data out there, because perhaps hearing that other people were going to be publishing something extremely similar, this is kind of the feeling that I get. I could be wrong, right? But I get a feeling that if they had more time, they would have looked at other organ systems in the mice, and they would have looked at other human cell types as well. Looked at replicative potential, and there's so many other things you can do. Yeah, John.
I found this fascinating, and I was trying to figure out. It seems like they did 3 injections in the mice, 2 different AAVs on day 1, and then doxycycline on day 2. And I'm wondering why they did the second AAV injection, which has something called RRTA, and how does that interact with the doxycycline that's injected the next day? Do you happen to know that very arcane technical detail?
No, I thought the doxycycline was just in the water.
Yeah, I thought so too. I think the doxycycline was in the water.
Oh, maybe it was in the water. Yeah, but
Yeah.
But why is there an RRTA AAV as well as the AAV that's carrying the 3 Yamanaka factors? Do you have any thoughts on that? It seems to interact.
Yeah, you mean versus having the transactivator in the same system, in the same virus? I'm not sure exactly technically why the system has to be split that way.
Is the payload size thing? AAVs don't hold a lot.
It could be the payload size issue. It could be a payload size. Yeah,
they are tiny.
Yeah, so you guys are probably right about that. That's probably a technical reason. That's probably just the most prosaic reason for it is they couldn't stuff everything in there into the vectors.
I think I'm not sure, but if you Google this RTA, it's a reverse tetracycline transactivator, and it seems to be helpful in the regulation. So maybe if you don't use it, you get teratomas more easily. Just a guess.
Well, I'll look into it further, but thank you.
Yeah, it could also be important for tighter control. Yes, Mika.
So there was one thing that I was confused about. In the methods, they talk about how they euthanize any mice that reach basically a certain critical threshold where the mice are just quality of life is deteriorated to nil. I was wondering, A, I'm guessing that's just for ethical reasons, or is there some other reason? And B, I couldn't find how many of these mice died because of natural death, and how many died because they're euthanized? Like, were they all euthanized eventually, or did some of them die before euthanization?
It's a good question. I thought I assumed, probably wrong, is that you just...
If you scroll down a little bit to the next subsection there.
Mouse studies and frailty scores, this one? Okay.
Yeah, in that section is where it talks about it.
Okay.
I think it's the second half of it.
Yeah.
Correctly.
Okay, euthanasia criteria. Rapid or sustained deterioration in health status resulting in body condition score of less than or equal to 2. Tumors or other masses become ulcerated or interfere with the ability of the animal to eat, drink, or ambulate. That can't be.
Yeah, and they didn't mention any mice getting tumors in the paper, but that'd be something very good to know. Like if any of the mice died from tumors, that seems important, particularly teratomas.
Yeah. That's a good catch. I think mice in general, and certainly rats, they tend to get a lot of tumors like later in their life cycle. That's like extremely common. But yeah, given the fact that teratomas are I think they mention here that they didn't, although they don't quantify this, they didn't mention there were no. Let's see, did they mention any? I think they mentioned, again, they don't show the data, but I think they mentioned that there wasn't more teratomas. I do know that they mentioned that somewhere, but it would have been, to your point, since teratomas are a consequence of, cellular reprogramming factors, it'd be nice to see that also parsed out, right? Like how many mice have been euthanized. You would assume that the euthanization would be, equal for both, and that it was whoever was running the experiments, that this was also blinded as well, right? So you don't kind of nudge yourself into prematurely euthanizing more mice in one group than the other, right? So so that is trying to see if
as this goes through the peer review process, they'll hopefully shore that up a little bit, and we'll see more data. I did read through the supplemental, and unfortunately, it didn't have details.
Yeah, right here it says we did not observe any gross terror, but cyclically these observations, in general, they didn't. I mean, they mention it, they don't quantify it. There's no pictures of it, so it's sort of one of these things where it's like, okay, well, we'll have to accept the validity of this claim. But yeah, so there's a lot of things that would be very good to have. And the the euthanasia aspect. I didn't actually know that, because I haven't done for myself. I mean, did lifespan analyses on yeast, right? So you just let them die. But I didn't know that. I thought with mice you basically just come in and find a dead mouse in the cage and you mark it as dead, right? You don't.
Yeah, I assume that
prematurely.
As well.
You don't prematurely euthanize the aged mouse based on ethical reasons. Actually, I don't remember if I actually read that in any other paper. Didier, you have a comment?
Yes, so first, like you said, yeah, it's good to see this time, let's say, normal mice measuring lifespan. So, and with a very good result. No, I saw a few people asking questions, so I will take the questions I read. I'm not convinced, but there were two people saying, oh, I couldn't see if there was really a control group. I mean, if it was not compared to an abstract group, but I would be very surprised. But just asking, there is for me no element going in this direction. That's the first remark, question. The second thing is why. So you will say I complain in the other direction now, but why did they take very old mice already? Because 2 years, it's in most cases it's 18 months. So here it was 2 years. I was kind of surprised. Okay, and so there were people saying yes, actually they show only the end of the curve, and this is very spectacular, but it can be almost by chance because it's actually a small part of the total life. Do you have comments on this? And yeah, no, on the positive side, yeah, it's great. It seems to be great, at least. There are many elements saying that it's great. Do you know how, let's say, easy it would be to do something similar to humans? Or just curious? Absolutely no idea.
Well, it's certainly doable in humans. I don't know what the side effects would be, and obviously you have to regulate the dosage very carefully. I don't, yeah. And whether or not this, whether or not you can apply this type of, again, it'd probably be easier to apply it to a specific organ, right? That's aged. If you want to rejuvenate, I think I mentioned this like in an earlier show, right? That I can certainly see this technology being used to improve the function of a failing organ, like a kidney or a liver or some other organ, where this individual is now at is now a candidate for an organ transplant because the organ is not able to carry out its functions, right? So if you can improve an organ using this technology, even doesn't even have to be like a fully rejuvenated, right? But enough to regain functionality where the individual now is off of a transplant list. I can see this being used and being kind of expedited for therapeutic purposes. If that even made even a slight dent in getting people off of an organ transplant list, right? So I can certainly see the technology being trials being done in that direction, and being expedited versus injecting it, completely in the entire, into the entirety of the body and seeing, like a kind of a full rejuvenation everywhere with all the kind of all the problems you might have of having, teratomas and other issues happening throughout throughout multiple organs or throughout the entire body. So I certainly do see that translatable, at least from reprogramming and individual organs as a first step. Maybe there'll be some other better technologies for this type of delivery, but I can certainly see the steps being taken in that direction. Why they waited so long with these mice? I don't know. They mentioned in the paper the reason they did it was that it was again, I'm just going by what the authors themselves have said in this paper, and that this would, delivering this into much older mice is a much more, how do they put it? It's a much more realistic test of the system than adding it, much earlier, right? Before, they could have done it when the mice were middle-aged, right? They certainly, rather than waiting at such an extreme age. So I'm not sure why they didn't maybe do a couple of age time points and say this is OSK injection for early middle-aged mice, middle-aged mice, and then very late mice, versus just jumping all the way to the very end of the life cycle of the mouse.
Yeah, because once again, I interrupt, because once again, when you take a first look at the curve, it's very spectacular. But when you take a second look, you see, they survived only a few weeks, all of them. So, something, once again, I don't get.
Yeah.
And, why not, better than other, but why not this 18 months mice like Aubrey de Grey is doing? And Aubrey de Grey was saying, these mice are really already very old mice, so you can, you are too late, but the results are there on the other side.
Yeah,
if they are not cheating, the results are really great.
Yes, and I agree with you. Like if we look at the curve, like I would think that the most optimal time to do this injection would be right at that point where you start to get, where basically aging starts to kick in on a lifespan curve, right at that point, rather than halfway down, right? So I would also expect there to be better results if you did it at that point as well.
And I'm coming to I'm thinking now about a simple other question. Is there do we have enough elements in the article and so on, enough information to try again with other mice? I think so. Huh? I think we do. It's transparent.
Yeah. I think we can certainly. I think there's enough background information here.
Because then my idea thinking growth, it is very important to start as fast as possible with mice of 18 months. To see what are the results.
John.
Yeah, my thoughts on that are that it's very good to start with the older mice because we're showing partial rejuvenation. If you start with young mice, you're not rejuvenating them, you're merely extending their lifespans. And related to that, as people get older, you can't say, well, we're going to go back in time and treat you as a baby. We need treatments for older folks that could partially rejuvenate them. And it was very interesting that not only was the remaining lifespan increased, but also the frailty index improved. So this really is indicative to me of a partial rejuvenation and not merely lifespan extension. And I'll just add that as soon as we can do it, I'm ready to volunteer to try the injections on myself.
Yeah. Well, I certainly think, and I hope that with kind of a lot of this data that's accumulating right now, that certainly people that are at risk of dying because of an organ transplant or lack thereof, that you could expedite trials for those individuals, right? Because if you're at risk of death, then the risk of a teratoma is pretty low, right? I mean, even if you do get a teratoma or some other tumors, if your organs are failing, then rejuvenating them and trying this technology, I think we'll see that type of application. And I again
related to that, Moderna and other companies are developing vaccines that will activate the immune system to wipe out cancers as soon as they start. So cancer might not be a risk that we need to worry about for much longer.
Well, I certainly hope so, right? That would be incredibly awesome. Yeah, Mika.
So just on John's point, my personal suspicion is that teratomas are not going to be tackled by the same tech that tackles most other cancers, because teratomas are kind of a little bit, as I understand, it's a little bit special, where it's basically your body has your cells have dedifferentiated and are now just growing what they think is the right thing in the right place. They're not the same. Someone correct me if I'm wrong here, but not the same as like a traditional cancer where it's just kind of run away. Some cell just reproduces really fast. And so I kind of suspect that the anti-cancer treatments we're seeing are not going to address teratomas, which is why teratomas showing up with a rejuvenation therapy are more concerning to me than if it was just like, oh, you get whatever normal cancer. Because I do agree we're probably going to have normal cancers solved soon.
Also, because they were only using O, S, and K and not M, there's less risk of cancer. I think that's why David Sinclair only used O, S, and K.
Mm-hmm. Yeah. Somebody on Facebook asks, Madonna Sechota, can you apply these factors to the eyes? Most likely you can, to get rid of macular degeneration and myopia. I have myopia. I'm not sure, because myopia is like your whole eye is like, and nearsightedness, farsightedness, right? So is kind of stretched out in a certain way. Perhaps macular degeneration. Yeah.
Isn't this exactly what David Sinclair did 2 years ago and end of 2020?
Yeah.
He did eye rejuvenation with OSK specifically.
Mm-hmm.
And Mika's beat me to it. I was just about to say the same thing. So they also crushed the optic nerve in that experiment as well, and they rejuvenated the eye using Yamanaka factors too. As to the eye shape, I have the same condition, so I would be interested in that. Maybe we can restore 20/20 vision with OSK.
I see a lot of glasses in this room. I think we all probably agree.
Yeah, I mean, I'm short-sighted. So I don't need to wear them for a computer, but driving or any distance, I have to wear them.
Yeah, I keep hearing, because I've been nearsighted like for most of my life, and I keep hearing that, hey, you get older and sort of as you're kind of your eyes get older, your nearsightedness improves, right? And I've just been getting more astigmatism. My eyes are just getting worse all around. I'm still nearsighted, but now I see like double vision from a distance. So I'm like, okay, I'm not getting the benefits of this old age eye reversal. It's just falling apart in general. So yeah, it would be great if this technology could keep up before I go blind. Yes,
so I had. So in the supplemental data that they supplied, they list a bunch of different metrics, presumably all the stuff that went into the Frailty score, along with weight. And one of the things that stood out to me was the weight of the treated mice appears to be at a human eyeball glance significantly higher than the untreated mice. And I thought weight in mice was usually the other way around. Like a rejuvenated mouse, you'd expect to get thinner. Is that correct? Regularly an aged mouse.
I think it varies. I think really old animals get really they lose a lot of muscle mass, and they tend to get a lot thinner. Yeah. So I think really old mammals just tend to get skinnier because of loss of muscle mass and less cellular turnover in general.
Also in that, the other thing that kind of stood out is inverse, and I don't know anything about mice, but they have, as part of the Frailty score, they include tail stiffening, which based on the numbers looks like tail stiffening is a bad thing, and that was the other thing that the treated mice seemed to get more of, whereas the untreated didn't. And part of the problem with the supplemental that hopefully they will get shored up when they get this peer reviewed. The supplemental doesn't say when they took these measurements for each of the mice. Was it like at the end of the life or at a certain point in time they took measurements for all of them? They just have measurements for each of the mice in the control and treatment groups. But I don't know, like, were they all the same age when they got this, or did they wait until, the mice looked like it was about to be dead, or was this right before they euthanized them, if they did euthanize them? So that would be nice to have a better idea of, like, what this data means, which is unfortunately missing.
Yeah, you're right about that. I'm going to give them based on how this data was presented, I'm going to give them a benefit of doubt and figure that they got some really kind of spectacular results with the lifespan of the mice, and they heard about other papers coming out like right on the tail of this, and they're like, well, we got to publish what we have and get this out there. I'm guessing that's the way it happens. So. Exactly. So they got this out, and now there's, like, to your point, there's all these points that need to be kind of like addressed. How many mice were, what were the exact criteria for euthanasia? They mentioned it there, but are we sure that it was, obviously you have to do the same thing for all the controls, right? Yeah. They have the historical data there, and again, I didn't know that for mice that was a common thing, that if you have certain infirmities of a mouse when you're doing longevity studies, that euthanasia plays a role in kind of culling them, so to speak, and putting them out of their misery.
Yep, it does, Oliver. I can speak to that somewhat because I worked with Alexandra Stoltzin when we were designing the protocols for senolytics, and there is actually a section about the euthanasia. So if one of the mice, for instance, is injured, because they do fight sometimes, and sometimes they injure themselves or fatally, and in that case, the best thing to do is to actually euthanize them for ethical reasons, because obviously they're not. It's basically putting them out of their misery. So quite often that is a thing with mouse experiments, that you do have to state under what circumstances you euthanize them. It's just one of those things.
Okay. Yeah. So yes, Didier.
Yes. First about this. Like Steve said, it's kind of an ethical obligation, but of course it's very complicated because the decision to euthanize is kind of a subjective one, and of course you will be tempted, except if it is blind, but I'm afraid the experiment is not blind. Except if it is blind, you are of course tempted to kill the mice in the control group and to keep the others. But okay, now I have two questions. Once again, it's really very interesting, and I'm enthusiastic. The first question is, do you know some of these people? Because I took a look at the names. And I think, Oliver, you said at one moment it was related to George Church, or did
he's a co-founder of this company.
Okay.
So I recognize Noah Davidson. I believe he came out of Church's lab, if I remember correctly.
Okay. Okay. And the last question, it's about what you said for potentially for humans. You can inject this in one organ without, let's say, contamination of the other organs. It's so easy. I know I have no idea about this, because for these mice, apparently it was, well, the whole body. I know a mice is smaller than a human, but for the rest, just asking, no idea.
Yeah.
I can speak to that.
Go. Yes.
So Youth Bio is another company in the space doing this sort of thing, and they are specifically engineering for organ targeting. So their goal is to do some sort of delivery vector, AAV, lentivirus, whatever, but the payload will only work in specific organs or specific tissues. And so even if it does go wherever, it will just be a no-op in most tissues, and it will only actually do its thing in the tissue of target. And the reason they're doing that is because we found that for partial reprogramming, the time it takes to partially reprogram before you hit pluripotency differs by organ. So I think the liver is one of the fastest ones. So it only takes like a couple of days, and your liver will become pluripotent. You start getting teratomas in your liver. Whereas there's other organs which takes like a week or two. And so by their idea is by doing specific organ targeting, they can then time it better. So they can say, okay, so we're targeting the heart. We know it takes this long before the heart starts to run into risk factors, and so we can do it, time it accordingly for the heart, and then deliver a different therapy for your lungs, which take a different amount of time, a different therapy for your liver. The advantage of that is you have a lot more control. The disadvantage, of course, is you need to deliver hundreds of therapies to target all the different tissues.
I just want to make a comment on the decision to euthanize. At least my experience in the Convoy lab at UC Berkeley was that the university veterinarian made that decision, and it was only made when mice were gone and in danger of dying soon anyway, and put them out of their misery. It's not like the experimenters chose to skew their statistics by killing off certain mice.
Yeah, that would certainly.
Sorry, Oliver, to stop you. But here, is it indicated if it was blind or not?
I don't think
normally,
so. No.
Yeah, that's also something, where when I was young, the principle to have the blind experiments was kind of, I don't know, one basic principle, and it doesn't happen anymore.
Yeah, I would assume, right, that probably the ideal way to do it is to have like a veterinarian or somebody who's like not running the experiment, basically making the judgment call as to which animal gets euthanized. So they just come in and they're like, okay, this one has to go, and then just and that way it's very you have it essentially blinded that way, and have it basically a neutral party that's doing that. That would probably be the best way to do it. I don't know what their protocol was, so I can't.
But still, the best way is that the people who are working with the animals don't know if these animals are in the control group or in the treated group. The issue with that also is it's very tricky, because if a therapy is working, this applies to everything else with yeast or anything else. If you actually start seeing an improvement, then you will notice that there is one group is performing better, right? So you might then inadvertently care more about one of the groups, than the other if you start noticing a difference. So even if it's initially blinded, that can break down if a therapeutic is actually working.
I would have assumed that in a blinded mouse study that they mix up the mice. So you just have like an array of cages, and some percentage are one, some percentage the other, and there's numbered, and you don't know which ones are which. So there's no ability to group them, so to speak. Well, I guess if some of them are getting their coats back and visibly like rejuvenated, then you lose the blinding, certainly. But I guess at that point, you solved your problem. You're rejuvenating mice. Good job.
Right.
On the blinding, there was a study a while back. I don't have a link. Sorry. Where someone was doing a bunch of different lifespan things, and they found that I want to say it was rats, but maybe it was mice. And they found that one of the rats that lived the longest was the one that was taken home as a pet. And it wasn't any therapy. It was just socialization and love, care, yada, 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 which aligns with humans, right? Humans tend to live longer when they have good social bonds, etc. But what's interesting there is if you have an unblinded study, it is very easy to accidentally mess it up. Even if you think you're treating the mice the same, just playing with them a little bit more may do the trick that pushes them over the edge. And so I agree that blinding is very important in these studies.
Yeah, as silly as it sounds, even talking to them, right? I mean, because they are social creatures, right? So
Exactly.
so I'm sure they pick up on certain cues that you think are irrelevant, but aren't. And then that there's a study done many years ago that where it's so obvious in retrospect that having a more an environment that's more cognitively stimulating with toys and whatnot in an animal's cage will lead to different outcomes. If you're using these animals in a psychological study, right? So they found out that that these control cages they had were just basically very Spartan environments and were skewing results because that wasn't really a psychologically healthy environment for the mice, and the more complex environment, which was the experiment, was the real control. I mean, so the animals really needed much more mental stimulation. So yeah, things can get tricky, and certainly social cues and the psychological status is definitely going to impact the overall health of an organism. So yeah, these things that we kind of consider to be warm and fuzzy will have an impact on the lifespan of an animal because of stress. And male carers, yes, somebody sent Adrian something about male carers affecting lifespan of mice due to stress. I don't know about the lifespan per se, but it's the mice were, I think, agitated. There was a study where they basically had there was greater stress for like the male mice because they sensed smelled hormones in human hormones in the sweat. And they tested it by having T-shirts, I think, from different participants, male and female, and there was the animals certainly were able to respond. I don't know what they used as a metric, but certainly they were able to tell the difference between male and female. That was statistically significant. So yeah, there's a lot of variables depending on that could skew these types of studies. Yeah, it's very difficult to control for certain things.
Yeah. So the most urgent thing is to do it again. That's for me very clear.
Do it again.
Very
do it
clear.
and do it on different
very
strains.
Clear. And coming back to there are all these companies with who are supposed to have billions of dollars, but nobody is doing it once again. Probably, as far as we know.
Yeah, repeat it middle-aged, repeat it at different time points, repeat it on different strains, and if these results are as robust as they are, seeing that you're getting a significant increase that late in life, then I would expect that we would have some spectacular data from having repeated trials across many strains.
Not really sure what strain you would go for though, really, because Black 6 is about as normal as you're going to get. Anything else is usually quite weird or unusual in some way. Any suggestions on strain? I mean, B56 is probably the most ubiquitous.
Probably different species. Those probably make more sense. Maybe like rats.
Rats, rats would work.
I think the other thing that I would like to see is more data on what the mice are dying from. I know it's challenging, but I would love to better understand, like, okay, so you kept them alive longer, but then did they die of the same set of things that mice normally die from, or did they die from a different set of things? And that would suggest that like maybe we are clearing one roadblock and there's a different roadblock ahead that we need to clear separately. And knowing what they died from would help tell us that.
Upper respiratory tract infections tend to finish a lot of rodents off. So it'd be interesting to see that data.
Yeah, the other thing that that occurred to me, and this relates to I think Oren, you're still here. Yeah, you mentioned the paper. We had a short back and forth on email, and that was you reminded me that one of the early papers we covered here was the one that looked at somatic cell mutation rate across species and how the rate basically is correlated very strongly with the lifespan of an organism. And these cellular reprogramming factors, right? They change the methylation status of cells. In general, I'm of the opinion that aging is basically a result of entropy, right? And entropy can have many different forms. You can have essentially basically a aberrant methylation status, but also an increase in somatic mutation, right? Which is basically the the root level of entropy for the genetic code and misfolded proteins and all kinds of other things. So loss of information. So we're getting this extension, right, with this technology adding in OSK factors, and you're getting partial reprogramming, and you're getting methylation clocks being turned back. I'm just wondering if you're also tackling some of the other, kind of entropic manifestations of aging as well, right? So I would think that you're probably not going to reverse some of the somatic mutations that are accumulating. So I'm wondering if, we could use this, we could see, how much of an aging reversal happens. And I would speculate that you probably need to have other interventions as well if you wanted to, let's say, completely reverse aging and say, okay, we're going to have a therapeutic approach that totally reverses aging, right? So you can, as long as we keep applying this, you live forever, right? As long as a rock doesn't land on your head, right? So you're basically you're just coasting along, and not aging. I would speculate that that there's probably additional things that we need to do, that perhaps these OSK factors are not tackling all of the entropic manifestations of aging, but a certain significant subset nonetheless, right? And so that was just a thought I had. So just
I would agree with that. I think the OSK treats epigenetic changes and developmental changes, but we've got the line 1 and other transposons that are actually causing more and more mutations with greater age, and I think that would have to be treated with something like CRISPR base editing or some other therapy. So I don't think there's going to be just one single therapy. I think we need to think about a collection of therapies.
And mitochondrial mutations, I think, is a very, very strong factor in aging. So like John said, it's going to take a multiple approach here. But yeah, epigenetics, I still maintain that I think it's going to be the biggest bang for the buck in the near future. And by near future, I'm talking 10, 20 years here, guys. That is near future.
Yeah. So yeah, one question that Keith had when we were kind of discussing this paper before the journal club was if that's the case, that it's not tackling all of the entropic conditions that lead to aging, then what you would predict is that if you do this type of therapeutic intervention, there's limiting returns, right? So if I injected these OSK factors and got partial reprogramming, and then let's say you extend X amount of years, if you do that later, then I wouldn't expect the same benefit, right? That you would get diminishing returns because you have other damage that's accumulated. Nonetheless, you still would get an improvement. That's a prediction I would make.
So, and something I would like to see in future studies that replicate this is trying this alongside other therapies like caloric restriction and the senolytics, the standard set of things. And I know there are people finally, after all these years, starting to do that. I'm hoping they introduce partial reprogramming into that set sooner rather than later, because I do think it's, like, I agree with Steve, this is going to, I suspect partial reprogramming is going to be a very big one. But, it may be that we don't see a lot of the benefits of some of these if there's some other roadblock that's right after it, right? Whereas if you can clear out the senolytic, do senolytics and partial reprogramming, maybe that clears out the two biggest roadblocks, and these mice live, twice as long or something. We won't know until we try.
Yeah, that's an excellent point that you raise, Mika. I've been talking about the internal factors that lead to aging, but the environmental factors as well, right? That could accelerate or decelerate, because these mice, I believe, are probably being fed ad libitum, right? They basically have just been, they get their mouse chow and they can eat at will. But that's not like, right? That argument goes back, for many years. Is that really normal, right? And you're doing your caloric restriction, and is that actually more normal, right? It's this equivalent of having more mental stimulation, having toys in a cage, right? So if you optimize the environment of the mice, if we calorically restrict these mice, do you see the same benefits of this OSK therapy, right? Or I'm not sure. I'm not sure. So if the mice are, if these OSK factors are actually tackling a fundamental facet of aging that's related to entropy, that relates to the methylation status, and that basically becomes aberrant, then I would assume that if you have calorically restricted mice that live longer, and then you put this intervention at the same equivalent point, that you would also see an extension, because you are again, reversing or resetting the methylation status. I would predict that, although obviously you have to do the experiments. So doing these experiments with those mice would also yield some very interesting data and some interesting conclusions as well. So yeah, there's a lot of potentialities for having variations on the experiments that were presented here in this paper. All right.
So I do have since we're on the topic, I have a project that I've been dwelling on for a while, and I just need to find some scientists to do it for me. But what I'd like to see done with partial reprogramming is, and maybe someone here can tell me why it's a terrible idea, but I'd like to see someone take a partial reprogramming, so OSK, whatever, and put it in the cell in such a way that it expresses until some new protein shows up in the cell that is indicative that the cell has reached a certain state that is a desired target. And then once that protein or mRNA or whatever shows up, then the therapy stops expressing. So basically you have so right now we have these therapies that are triggered by doxycycline, right? So you add doxycycline, then it starts expressing, and then take doxycycline away, and it stops expressing. It feels like the better way to go here would be to have a therapy that expresses until the cell reaches the target age and then stops expressing. And you detect the target age by detecting, there's a protein that's now in the cell that doesn't usually show up in old cells. It only shows up in young cells. And so that's when you know you've reached the target, and you turn off the therapy when that's present. Or even better, destroy the therapy. So delete it entirely, so that way you don't have to worry about it overexpressing. I'm curious if anyone has thoughts on that.
Go ahead.
There was an experiment a long time ago in a galaxy far away by Jean-Marc Lemaitre, the guy from INSERN who's done a lot of the early OSKM work, and I'm pretty sure it was Lemaitre. He showed that there's almost like a natural sort of buffer, I'm going to use that word, for when you partially reprogram a cell. This is before it loses its cell identity and becomes a fully reprogrammed cell. There seems to be a natural buffer whereby cells, sort of regardless of how old they are, because he took them from a range of people. I think there was somebody like 106, 80-year-olds, 40-year-olds, and they rolled it back. And it seems to sort of default to around biological age of 25. That seems to be the sweet spot. So there's got to be some kind of markers or something that occur at that point to tell it to stop. Otherwise, how is the buffer created? So it's interesting that irrespective of the age group, it always seemed to reach that sort of sweet spot. And I think it's been vindicated fairly recently, maybe by Sinclair, or it could be Belmont. Ocampo and those folks who, of course, they now work at Altos Labs. So I think it's definitely coming, but there does seem to be that sort of Goldilocks zone for partial reprogramming.
So Mika, while you're mentioning that, I just had a thought. I don't know if anybody's done this or if it's possible or if it's technically feasible, but obviously one of the metrics that we're using here is change in methylation status, right? So you get back to a more youthful or normal methylation status. So to your point, I wonder if you can make a synthetic promoter that basically acts as a kind of as a feedback system for switching on and off the OSK factors. So basically, the synthetic promoter would have methylation sites that correspond to methylation sites that get changed by the OSK factors. So as you increase the OSK factors, that promoter is switching on the OSK factors. But when the methylation status gets to a certain state of that promoter, it switches on a repressor that then shuts off the OSK factors, right? So you basically, the OSK factors themselves are regulating their own expression based on methylation status of a system that regulates them. Does that make any sense? Am I making?
Yeah, and I think that would definitely be ideal. If we could have these, so inject them once and then it would self-regulate from then on.
Right.
That'd be awesome.
Right. So if there was some kind of synthetic methylation, some kind of synthetic site that basically could respond to the changes in the methylation status, and then you can program that site somehow to then be able to toggle on and off the OSK factors. Then to your point, Mika, you can just inject it, and it would just sort of auto-regulate itself, right? So that's I don't see technically why that's not possible. You can certainly play around with this system in vitro at first, right, and put it into cells and see whether or not you basically have this sort of you can create this sort of auto-regulatory system that would then optimize the dosage of the OSK factors, right?
Auto-regulation is an intriguing idea, but that assumes that the automatic cutoff point is known in advance and correct for everyone. So I think if it were me, I would prefer to have manual intervention and know when I need to drink my next doxycycline soda, and then wait until I need to do it again, rather than trusting that the thing that was injected in me 3 years ago is still going to be auto-regulating correctly. That's just a personal thought.
Yeah, well, the synthetic biologist would love something like that, right? Because that's what synthetic biology is all about. Different ways to synthesize DNA, but also different genetic circuits that can be cobbled together to basically to do cool things inside of a cell. So, if I had the time and the money and the lab space, I would after this call, I would go in there and start trying to build that kind of circuit, and see if we can get it to work. Yeah.
Just to say, it was very interesting, but I have something else where I have to be in 17 minutes. Goodbye.
Okay.
See you. And maybe, Oliver, I will take you small contact again to continue. I brainstorm about this. Thank you again. Goodbye.
Thank you. Yeah, so a lot of cool ideas that this paper has kind of sparked amongst us, I think.
No, thank you very much. I also have a meeting that I'm late for. Appreciate this. It's a lot of fun.
All right, I think that wraps it up. If anybody has any other comments, I think we've wrung out as much as we can wring out from the data that we've seen here.
Exciting stuff, guys. So
and a stimulating discussion as always.
You wait till next week when we're Next week, good grief. Next month, get a hold of yourself.
This is going to be a daily show. We're going to read one paper every day. We're just going to plow right through every. No.
Yeah, you know that can't happen. So I'm going to tentatively say that we might be talking about viral vector delivery of collagen producing triggers next time. So in other words, can we do in mice in humans what they've already done? So that's pretty interesting. So yep, kickstarting collagen production in cells. We touched upon it briefly before we actually went live today, but a recent paper's caused quite a storm that they managed to actually get viral vectors delivered to aged skin in mice, of course, always in mice first, but it does actually start collagen production, much like they are in happens when you're younger. So that would be cool, kind of like a gene therapy version of Botox or fillers, and it would be totally natural. So I've always said though, skin, it's the most obvious choice for the things that we do our field to focus on first, and people are very, very focused. The public are on appearances. We all like to look pretty, I'm sure. So I think that's why it's grabbed so much attention, and I reckon it could be a good topic for next time. Let's talk about skin rejuvenation via viral vectors. I reckon, Oliver.
Mm-hmm.
Well, that's what I'm gonna suggest for next time, and I think that will spur some quite interesting discussion. I mean, we all like to look pretty, right?
Right. But at the end of the day, we should all, heed Mr. Rogers' advice and be happy with who we are.
I don't know. I think he sounds a bit of an apologist, aging apologist. I do like him, but he was very cool. But I'm not sure he would even agree with that, Oliver. He'd probably say, "No, hell no, I take the gene therapy."
Might be right about that.
Yeah.
Anyway.
Well, so yes, we will see you all next time then, and I'm sure it'll be either that paper or something equally exciting. And let's talk about that next time. And thanks to everybody that joined us today here, and also out in Facebook land, where there are some other folks watching. And thanks obviously to the heroes who make this show possible. And if you'd like to learn more about that, you can visit our website, lifespan.io/hero or heroes. They both work. So we've got all bases covered. So I guess we will see you next time.
All right. Take care, everybody.